packet, it follows along with the same exact questions. So we can do those questions that we didn't do in class as well. So this one has our expected versus our observed numbers. Usually when we look at genotypes, we can look at the three genotypes and we might think, oh, the degree of freedom should be the number of categories minus one. And that is true, right? So oftentimes, if you are just looking at the three traits, it will be 3 minus 1, and the degree of freedom will be 2. However, in this particular question, it's actually like it didn't give you the entire background knowledge on the question. So this is like not a full question. But if it were to be a full question, it would have said, we want to do a chi-square analysis to see if the population is in Hardy-Weinberg equilibrium. So in that case, we're not just talking about the genotype and the characteristics anymore. We're talking about the P and the Q value, like the allele frequencies reflected by the genotypes rather than the genotype itself. So you will still do the calculation the same way of like setting it up with the 10 observed minus expected squared divided by expected, add it all up, get this number of chi-square, but in your interpretation, the p-value of 0.05, Toby, come here. The degree of freedom will actually be 1 because of the Hardy-Weinberg equilibrium. Like we really have two variables of the dominant versus recessive allele frequencies rather than thinking about the three characteristics. So the calculating the chi-square value is fine. Writing out your conclusion, because we're not just writing out big rejecting the null hypothesis, the population is not in Hardy-Weinberg equilibrium. You got to say what p-value you're choosing and what degree of freedom you're using. So that's a little bit of a tricky part. So in this case, the critical value is 3.84. This question could show up or not, but if it were to show up, it could be, if it were to be an FRQ question, then you get a chance to answer those different parts, and you would probably just get a point for this calculation alone, and then get another point for maybe the conclusion. Or this question could also show up as a multiple choice question, in which case, like it could just straight up ask you, what is the critical value? And then you won't even have to do the calculation, you just look at the chart. And then you have to find the right critical value, which is where like kids will make a mistake, and it will be a question where most people get wrong, but now maybe you'll get it right. If that makes sense. But let me know if this doesn't make sense. I'm happy to explain it some more or explain it in a different way.
I have a question about it's like the p-value stuff. So we use the critical value to see if it's statistically significant.
Correct.
So if the chi-squared value is greater than the critical value, we can say it's um, statistically significant.
You can say we are 95% confident that we reject the null hypothesis, which means, like you gotta elaborate, which means this population is not in Hardy-Weinberg equilibrium. So it's also like it's all significant. Like it's all the same kind of confidence interval. It's just a matter of, are we 95% confident that we fail to reject the null hypothesis? As in, like we can't rule out that the population is in Hardy-Weinberg equilibrium, or we reject the null hypothesis and say the population is not in Hardy-Weinberg equilibrium.
Okay. And then for the degree of freedom, what do we use the degree of freedom to find?
You use it to find the critical values. Let me show you. Let me pull up the equation sheet so you see what that looks like. Equations. So this is what it looks like. I'm not 100% sure what this looks like on Blue Book, hopefully I can remember on Friday, or if I don't remember, you can do this on your own. You can do like a test run of the question of like some sample questions on Blue Book for AP Biology. So then you can test out like the different buttons. What can I do? How do I eliminate answer choices? What does the equation sheet really look like? Like, do you go into it and choose the equation? I think that's what it is. But anyway, the old equation sheet with all the equations, this is what it looks like. But the formatting is going to be a little different, but the information will be the same. So the degree of the degree of freedom and the p-value is what helps you find the critical value that you will compare the chi-square value to. So if the chi-square value was bigger than the critical value, so in this case, like the critical value that you will compare to is 3.84 because the degree of freedom is 1. You can actually, like you're giving this information as well, degrees of freedom are equal to the number of distinct possible outcomes minus 1. But it's just like with Hardy-Weinberg, you have to know that the possible outcomes is really 2 because we have the dominant versus recessive alleles. So then the degree of freedom is 1 instead of 2 if we were to just look at the 3 characteristics. Yeah. So it is important, like in your conclusion, it's really important for you to say, with a p-value of 0.05 and a degree of freedom of 1, the critical value is 3.84. Because my chi-square, you can see the conclusion. One more time. Like, you won't have enough time to write every single one of these words, but you do need to include all the components of like what values you're using, what you're comparing. Do you reject or not reject null hypothesis? And what does that mean? Yes, yeah. That is the most thorough answer. I would say, like, if the question says, like, what is the conclusion as an FRQ, rather than explain, then you might be able to shorten it a little bit and just say the population is not in Hardy-Weinberg equilibrium. Like, if it doesn't say, like, evaluate or explain your conclusion, then just say it's not at Hardy-Weinberg equilibrium will be fine. But if it does say, like, elaborate, like, evaluate, or it says, like, explain, blah, 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 then you need to write the whole thing. Yep. Any other questions about this?
No, I think I'm good.
Okay, great. All right, so moving on from here, with this unit, we need to know all these different things, mitosis versus meiosis. We just reviewed that, so we're gonna probably, it'll show up again. We'll just take a very quick look as a refresher. Mendel's laws, the different ratios that we talked about in the past few days, how to do a mono hybrid Punnett square, how to do a dihybrid, test cross, all those things. Different types of crosses, X-linked, there really like is a lot to it. You need to be able to do every single one of these examples to be fully prepared. And my suggestion really would be, back in the days, we had like the big packet and the small packet for genetics. Just going back and maybe do like 1 or 2 of those practice problems for each type of the like genetics. Because this is really just like math, like you practicing it is what's going to get you comfortable. But we can obviously talk about the basics and do one problem together for most of these categories. But I would say like over the weekend, if you have time, just give yourself the proper refresher of these questions. Or another idea is you could look at the old test questions from Unit 5, and redoing those questions are really good practice as well. Because then those are actual like, yes, you have the skills on how to do it, but these are how the questions are worded. Either one is fine, but you just want to be like, you know how to do it for the most part, but you knew it really well like a month or 2 ago, but now it's less fresh. These are questions we talked about in class, just some like review questions for meiosis. So I'll just click meiosis and some of the other topics too. I'll just click through it real quick. You can find these slides on Veracross. You can go over it yourself. Or if you like catch one right now, you're like, wait, what is the answer of that again? Then we can go over it, okay? So I'll just leave it up here for a minute so you can skim through it and let me know if you would like me to talk about any of these questions. Minute is really long. You, okay. I'm gonna click through it, okay? I think there are quite a few pages. Here's another page, mostly comparing the differences and similarities between mitosis and meiosis. You can talk about the number of daughter cells. Are they identical or not identical? Genetic components of the daughter cell versus the parent cell. How many times the cell is dividing during the process of mitosis or meiosis. Whether or not crossing over or independent assortment takes place. Whether or not there is genetic diversity. So those are all differences that you can say. Next one, meiosis again. So we just did this. Should be okay. I think. Yeah, the highlighted ones were the ones that we talked about in class because those were the slightly harder question. Do you know the answer for number 4 though? What process during meiosis 1 that causes daughter cells to be haploid? It would be
Cytokinesis.
cytokinesis, exactly. That's right. So all the previous steps just really get you to that point, but cytokinesis is what actually cuts the number in half. Oh, this is a good place to plug. This is really a unit 4 question, was like the cell cycle. Mitosis and meiosis are a part of the M phase of the cell cycle, but the M phase is really divided up into mitosis followed by cytokinesis. So it's like G1, S, G2, and M. That M really stands for mitotic phase, which includes mitosis and then cytokinesis following it. Yeah, it's just interesting. Mitosis and cytokinesis really follows mitosis, and meiosis has cytokinesis following each step of meiosis as well. Okay, on to the next. We talk about these questions: common ancestry, ribosomes, Mendelian genetics, thinking about how like genetics really is related to common ancestry as well. Metabolic pathways. Oh, laws of probability. The multiplication rule and the addition rule. So what you see over here with the blue box, that's given to you on the test. So if you understand what it's trying to say, like A and B are mutually exclusive, then it actually tells you like A or B means plus, A and B means multiplication. So that's really good. Yeah. So let's say if you're thinking about an offspring that has like brown hair and curly hair, but also big nose and like freckles, and also I don't know, a really long arms. Those are all those characteristics in one organism. So that's the and you want to like, you can calculate the probability for each of those characteristics and then multiply those together. We'll see an example of that, I think. The A or B will be like, you have 2 children, one of them has brown hair, or you have 2 children. What are the chances that one of them has brown hair, but the other one has black hair? So then you would take, wait, no, that doesn't make sense. No, that's also multiplication. What are the chances that your first child is colorblind or not colorblind? So you will do like the probability plus the other probability. Yeah. Another example of, let's see, another example of A or B. Ooh, this is like, if they are really trying to scam you with the question, they can put the and the or in the same question. It could be like, what are the chances that the first child would have black hair or brown hair, and the second child will be colorblind or not colorblind? So then you will like, for the first child, you will do the like the 2 traits. So then it will be the multiplication rule, and then you do the multiplication rule again separately for the second child, and then add the 2 together or something. Or the other way around. Anyway, look at the questions very carefully, figure out which part is really the and, which part is the or. If it's happening at the same time as an and, use multiplication. And it tells you and means multiplication. It's just making sure, like reading the question carefully and understand what is the question trying to tell me. Question number 9 is worth talking about. What is the probability of maybe in a tetra hybrid cross? Actually, without giving the parents, I guess without giving the parents, it's hard to do this. We'll do this later. Sex-linked traits we talked about in class. Let's just look at examples. Do remember, once again, emphasizing phenotype is about genotype and the environment, and natural selection interacts with phenotype, which is again influenced by the environment as well. This is interesting. What is the something called phenotypic plasticity? Like your phenotype could change during your lifetime as well, which is what oh, or like B, for the second question, environmental factor affecting gene expression. So in A and B, these two pictures, we're really looking at caterpillars. And depending on like, are they living on the stem part of the plant, or are they living near the flower part of the plant? Then they would actually like their gene expression patterns are altered, and like what they look like will be different. They have different camouflage. Also on the right side was these butterflies. This is the phenotypic plasticity, where the phenotype actually changes during its lifetime or during different seasons as well. Just so interesting. Look at these butterflies and the colors. Wow. Okay. Next one, chromosomal inheritance, relating to non-disjunction. So like the law of segregation is really like homologous chromosomes separating or the sister chromatids separating. And if that does not happen, then the daughter cells will end up with an abnormal number of chromosomes potentially. You don't need to know the exact genetic disorders that's shown on this page. You can think about how fertilization leads to genetic variation because it combines genes passed down from two different parents, which are like indirectly passed down from their grandparents and their grandparents and so on. So every generation you reshuffle the gene. If it's sexual reproduction, you reshuffle the gene, pick half of the genes, pass those down to your child, and then the other half of the gene that were also reshuffled get combined together through fertilization. Okay. Mitosis versus meiosis. Do you feel like you need to take a look? I'll just leave it up here for a minute, for a second. You can take a look at it. Let me know if you want me to talk about it, but it should be pretty straightforward, especially mitosis. But it's still very important because it does give you, oh, this is worth talking about. The role of mitosis in organisms will be asexual reproduction, actually, for some unicellular eukaryotes, and growth and repair for multicellular eukaryotes. Notice I really emphasize eukaryotes because prokaryotes, to make identical cells and reproduce, they go through binary fission instead. And they don't have the nucleus, so they don't need to break down the nucleus and reform the nucleus either. So that would not be mitosis at all. So question definitely can ask you about the role of mitosis in organisms. Ooh, it could even ask you, this one's kind of high tech, a little more advanced. Why do chromosomes need to condense during prophase? Essentially, well, the sister chromatids are held together by the centromere, and if the sister chromatids are condensed, then it's much easier for the chromosomes to align at the metaphase plate. So then each half of a duplicated chromosome can evenly divide into the two daughter cells. Okay. And what else? What else is advanced? The centromere's role, well, you can talk about the centromere's role is to hold the two sister chromatids together. The centrosome's role is to make spindle fibers, yeah, which are used to move the chromosomes around the cells and pulling the copies of the chromosomes or copies of the DNA apart from each other. Why do DNA need to uncondense? Because in order for the DNA gene expression to take place, the DNA needs to be uncondensed, like at certain parts, to then allow the enzymes to access the genes and be able to go through the transcription and translation later on. Okay, on the bottom, it shows you the mitosis versus mitotic phase. Here's meiosis, the whole thing. You can take a quick look and let me know if there's something you would like to go over. Also think about if I have any high advanced questions to ask. Okay, let's see. I guess I do want to emphasize over here again how crossing over takes place. It's within one organism. So it's pretty easy for people to make the mistake when we're doing those linked gene questions and go, wait, aren't they supposed to cross over? And they are supposed to have all these gametes? But you really look at, like, you have to look at one parent at a time and like not thinking about what's going on with the other parent, because crossing over is during meiosis of one parent. So just how the two chromosomes separating from each other before they combine with whatever is going on with the other parent. Like this chromosome is not going to cross over with that other chromosome that it's combining with. If it is producing another generation, then they can cross over. But like before fertilization, during meiosis, it's just one parent at a time. Um, just something to be careful with. Meiosis 1 and meiosis 2, diploid, haploid, haploid, haploid. Diploid having homologous chromosomes, haploid doesn't. So haploid independent assortment, like you have the maternal chromosome and the fraternal chromosome. During meiosis 1, the two separate, so how they align at the metaphase plate and how they assort into like these two chromosomes, maternal, paternal, their assortment is completely independent from the assortment of these two. So you can say like the two chromosomes move and like the genes get passed on to the gametes independently of the other pairs of homologous chromosomes. Something like that. Okay. That's meiosis. Any questions about meiosis? Okay. Diploid, haploid, we saw all this. Somatic cells and gametes. Somatic cells are body cells, gametes are gametes. All the rest of the words are good. Just a comparison chart that we did in class once upon a time. You should be able to answer it with no problems. Yep. Next one. Here's another summary slide comparing the two. Anything important that we did not talk about? Let's see. I guess number one is kind of interesting, saying like, when does DNA replication take place? Obviously, it's before the whole process, but I guess it's interesting to think about how it doesn't happen in between the two processes. The DNA is already replicated before all of this happens. Anything else? Oh, I guess for meiosis, it would be cool to talk about how not just the daughter cells being different from the parent cells, every single daughter cell is also different from each other due to both crossing over and independent assortment. So like if there was only independent and not crossing over, some of the daughter cells could be identical, right? Because like that's during the second meiosis 2, the sister chromatids separating from each other. So the sister chromatids were supposed to be identical. They really look like this. So then like the two cells that like end up from the initial cell at the beginning of meiosis 2, those two cells will be identical. But because of crossing over, the sister chromatids in meiosis after prophase 1 will no longer be identical. Okay. Mitosis. Earlier I was making second something I didn't talk about was with plants. Actually, plants can also use mitosis to reproduce. So like some plants can just like have new roots and grow a whole new plant out of itself. So mitosis is not just reproduction for unicellular, whatchamacallit, unicellular eukaryotes. Okay, these questions we already talked about in class. So we're gonna move on from there. Mendel's law of segregation, once again, is just the chromosomes separating from each other during meiosis. Like the homologous chromosomes separate from each other, ending up in two different or four different gametes. We're not considering, like, even with one pair of chromosomes, like just one trait, yellow versus green, there is law of segregation. You only end up with either big Y. In this case, it will either be this big Y or this big Y in the gamete. But then for the next generation, the gamete will either have, like, you will still have four gametes, but a gamete will either have big Y or another gamete will have big Y, or the other gamete will have little y, the other gamete will have little y. So really relating that concept of, like, why do we do a Punnett square? Why do we separate the two letters? It's because of the law of segregation. And then the Punnett square also shows you the process of fertilization, where you get this one gamete as a possibility, like 50% possibility from this parent, 50% possibility of the same parent in this case, or like a different parent but was the same genotype. And then the two gametes come together, fertilize, and give you the next generation with this particular genotype. Do be careful with genotype versus phenotype. The ratios are different. What else? I think that's it. Okay, next one. Oh my God, this dog. Toby. He only knows the sound of the treat. Just have to bring him over so he's not annoying. Okay, this next one is law of independent assortment. This one, you can't just consider one trait anymore. You have to consider two traits, two genes. And reminder, a gene is a sequence of DNA that codes for a protein or a trait. So if you are thinking about a physical trait, it should be at least related to one gene, if it were to be a single gene trait. The polygenic trait doesn't really work, like, for these Punnett squares. Anyway, so also for independent assortment, this only works if you're looking at two genes, two traits on two different pairs of homologous chromosomes. It also doesn't work if the two genes are linked on the same chromosome. In that case, like if big R and big Y are on the same exact chromosome, they're gonna go together. And there's the crossing over, but they're still, like, whatever is on the same chromosome, they're gonna go together. Anyway, law of independent assortment is what also ensures that every single gamete is different from each other, which increases genetic diversity, which increases the diversity of a population, which increases the survival of the whole population with environmental changes or without. It's just like natural selection. Having genetic diversity allows opportunities for organisms to survive. Okay, so different alignments of the chromosomes. We can have this particular orientation where the paternal chromosomes are on the same side and the maternal chromosomes are on the same side, gives you these two possible gamete outcomes. But it is still four gametes at the end of meiosis. Or with this particular orientation, you will get these four different types of gametes. So together, there are four different types of gametes overall, as well. So you can do your FOIL method here if you haven't done that for a while. If you do like big R, big Y, little y. Do you need to see the FOIL method at all, or you're feeling pretty good about that? FOIL? No FOIL? Sounds like no FOIL. Sounds like you know how to do it. Okay. Well, and also FOIL only really works for a dihybrid. So remember the other day I was drawing brackets on the board with, like, here are all the possible gametes. Drawing the brackets works much better for more than two genes. Okay, law of independent assortment, again, you can do your 4x4 Punnett square if you really have the time for it, or you can really just do like percentage, like the frequencies. Like if you're just looking for big Y, big Y, big R, the chance of getting big Y, big R as a gamete is 1/4. The chance of getting big Y, big R as a gamete over here is also 1/4. 1/4 times 1/4 gives you this, which is 1/16, and it is 1/16 of this 4x4 Punnett square, if that makes sense. Or you can look at one trait at a time. You can look at like big Y, big R, little r. Let me just draw this real quick. Where's the whiteboard? Oh, right here? Nope. Can't find the whiteboard. If you did like big Y, big R, crossing was big Y, big R, and you're looking for the big Y, you can look at just big Y's and think about, okay, if I'm just doing big Y, crossing with big Y, and I'm looking for big Y, what is the chance of having that child? It's 1/4 because of the 1:2 to 1 ratio. So homozygous dominant should be 1/4. And then same thing for big R. So it's 1/4 for the big Y, 1/4 for the big R. Together, that's 1/16 with the multiplication rule. If that makes sense, or if you need help with that, I can go over that again. Do you need to do this question that we went over in class one more time, or you felt pretty good about this particular example? This is just crossing a dihybrid cross. Are you still there?
Yes, sir.
Yeah?
Yeah, I'm
Um,
here.
Do we feel good about this question? This was from,
Yeah, we can go on. I'm good.
okay, all right. This picture shows you once again the here it is. Once again, the linked genes and how it doesn't follow the independent assortment rules. Okay, and then we have just another example of pretty similar to the one that we did before. Very quickly looking at this, it's asking you what fraction of the puppies are expected to have yellow and straight fur. So yellow in this case should be recessive because all the first generation have black fur. And then the straight fur is dominant. Okay, so if you were to write this out, it would be, is there a way that I could draw this? No. Let me see. Hold on, give me 1 second. Let's see this real quick. Way that I can draw this. Just do this. Yeah, just want to use this one as an example so you can see how this is, we can use the multiplication rule here. Okay, so for this one, you could do, so we know black is dominant, so you can do like big A for black, and then straight fur is dominant, so big B for straight fur. So we're doing true breeding, so that means these are homozygous, so all the F1 generation will be big A, big B, little b, crossing with another big A, and then big B, little b. It's asking you, what are the chances of finding a yellow straight fur puppy, right? So in this case, if you were to draw a 4x4 Punnett square, that's going to take you a while. And during the AP test, we truly just don't have the time for it. Or, I mean, if you do end up having the time for it, feel free to do it. But there's really a much easier way to do this question, is to just think about like one trait at a time. So if we're thinking about just the A crosses, right? We're crossing big A with another big A, yellow is recessive because yellow was the trait that got hidden away and then showed back up again through these homozygous crossings. So that means yellow has to be a. That's what we're looking for in the puppy. And what are the chances that we can get a a from our 1:2 to 1 ratio that we know for the heterozygous crosses? We will know the homozygous recessive is always 1/4. Okay, so now we're done with this. We can look at B. For B, we had big B, little b, crossing with big B, little b, so we can use the same exact ratio to work on this. But we're looking for straight fur now, and straight fur is dominant. So we're really looking for a big B with an unknown. So like it could be big B, or it could be big B, little b. But either way, you're thinking about the characteristic, the dominant phenotype. So this is our like this part of the ratio. Or if you think about like the 3-to-1 ratio of the dominant trait versus recessive trait in a heterozygous monohybrid cross, then you can also just use this directly and go, oh, that's a 3/4 chance. So now putting it together, we're looking at a puppy that is yellow and straight fur at the same time. We put it, well, you wouldn't put a cross over there, but it's an and situation, right? So that will give you 3/16. Okay. Yeah, and that's the answer. Questions about this? So this is you should be able to do this pretty quickly. We can even look at a bunch of traits, like you can look at 3 traits, 4 traits, 5 traits. We've done that when we were learning the unit. You just do that a bunch of different times. But it is important to think, oh, if I'm looking at a dominant trait, it could be homozygous dominant or heterozygous. You can just do like B and whatever the other thing could be. Now I don't know. Oh, there we go. Okay, questions? We're good. We're good. Test cross is always between an unknown. So this is really your, like we have a parent, we don't know what the second one is. It could be either one. We don't know which one it is. We always cross it with a homozygous recessive on purpose. So this is like intentional. We also find this goat intentional because we're like, oh, sheep. You're like, what is this sheep? What is it supposed to be? Well, through the test cross, you look at the babies. If all the babies show the dominant trait, that means the unknown parent was this. If all the baby or half and half, then that means like for it to have half of the offspring to show the recessive trait, that means it had to have gotten a little a from one parent or B in this case, and then the other little b from the other parent. So that means the other parent, the unknown parent, has to be heterozygous. So you notice like I am saying a lot of words. Maybe you already know how to do the test cross, but it is perfectly a fair question if they were to ask you to explain why, like how did you figure this out? Okay, so do practice or think about like how would I explain this? Think in terms of, oh, an offspring has 2 alleles, but 1 allele from each parent. So that tells me something about this parent versus the other parent. We did this question during class. Do we feel good about it? Do we need to do this again? Let me know if you need to do it again. I'm gonna scroll through it. Unless you need me to go back and just let me know. We filled this out during class, all these ratios, you just gotta remember it. Do pay attention. Is it phenotype or is it genotype? And is it 1 trait or is it 2 traits? Offspring ratio, here are the ratios again. Oh, what is this? I don't think we did this. Do you want to do this question together as a practice? Or do you want to move on and do a different type of practice?
We can do it.
You good? Just do it real quick? Okay, we can do this real quick. Okay, so this one, we have goats. With the goats, are you seeing the, hold on one second. You're not seeing the sharing screen bar, right? You're just seeing the slide. I just want to make sure I share the screen the right way. Yeah, we're good. I think we're good. Okay, anyway, what's the goats? Okay, so we have true breeding, once again, homozygous goats. We got white goat with short hair. We got brown goat with long hair. We don't know which one's dominant, which one's recessive, so you want to look for that information. Brown hair with short hair with all our this dog, with all our F1 generation offspring. So what that tells me is brown is the dominant one. So big A is brown. And then little a is, what's the other color? White. Okay. And then because all the offspring showed the short hair, that means the dominant one is the one that overpowers, like covers up the other, the expression of the other allele of the gene. All these fancy words, right? The expression of the gene, like what is that color really is going to look like? Or what's this trait going to look like? The expression of the allele, like that version of the gene. So those are all words that you can read and use on your test. We have big B for short, because that's the one that showed up. and then little b for short, and what's the other one? Long, I guess. Okay. So first it asks you the parent's genotype. That's super easy. It's just big A. Oh, wait. Oh, actually, once you figure this out, then you look at the parent's genotype. The white short goat is little a. Like, just because it's true breeding homozygous doesn't mean they have to be like dominant. That's two different things. It just means homozygous. It could be homozygous dominant. It could be homozygous recessive. So little a, and then it's short big B, big B. Crossing with the other parent, we have brown, which is big A, and then what are we looking at? Brown and long, so little b and little b. Those are our parents. Now, and that's it. That's it for A. B says we're crossing two random goats. We really don't know what they are. She forgot to look at the parent goats. So the offspring generation has three, like this is the offspring generation, right? We have a 3 to 1 ratio. You look at it and you go, what are these numbers? Well, looking at four numbers is really hard. It's easy to just look at one number. So let's do that. We're seeing that there are 3 brown and another 3 brown versus 1 white and another 1 white. So if I were to just look at the A trait, I'm noticing that I have 6 brown goats in the offspring F1 generation, right? Like the parental generation is really like we don't know what is going on with the parental generation, but we know F1 generation, the ratio is a 6 to 2 ratio, which I can simplify to a 3 to 1 ratio. And hooray, I know exactly what 3 to 1 ratio means. It means the parent had to have been heterozygous. So it had to have been big A, little a, big A, little a. You blindly grab some goats, but now you know what they are. Okay, then we look at the other trait completely separately. Okay, we see that we have 3 long hair goats and another long hair goat. So that's 4 long hair goats total. We see that there are where's it? 3 short hair goats as well, and then another 1 short hair goat. So that's 4 short hair goats as well. So we see a 4 to 4 ratio, which simplifies to a 1 to 1 ratio, and we love that. A 1 to 1 ratio for this other trait that we're considering, the B, the short versus long, well, that tells me it's a test cross, and it's a heterozygous and a homozygous cross. So we know one of them had to have been this, and the other one had to have been this. Okay, and because the first trait is exactly the same, that's all you need. That's the answer. But let's say in another scenario where, like, let's say what you ended up figuring out was like this and then this. No. Oh, sorry. This and then this. But then the other trait is big B, little b, and also little b, little b. This one's a little trickier. That way, there will be 2 different answers. You can have this combination, or you can have this combination. So like the A trait and the B trait, you swap it, right? You notice like this crossing is different from this crossing, but because like it's still a test cross, still a test cross, still a test cross, still a test cross, it will still give you the same exact answer. So in that case, there are 2 possible outcomes. And if it were to be a really mean FRQ question, you need to write the 2 outcomes. If it were to be a really mean multiple choice question, you need to account for both of these outcomes, which is you know, tough out there. That's why maybe an 80% on the AP biology test accounts for a 5, but the number changes, so you never really know. Yeah. So like if this were to happen, this would have been a this or that situations, you have to add the 2 numbers together, whatever numbers you get, if you were to do a prediction of like the probabilities and whatnot. Okay, any questions about this question? This is a really good example.
Yeah, I understand. So test cross means it's heterozygous and recessive.
Not
Homozygous.
necessarily. So I'm being careless about that. Like when I say test cross for the 1-to-1, I'm referring to the type of test cross with the heterozygous and the homozygous recessive. But what a test cross really is that I don't know if it's homozygous dominant or homozygous recessive. I just cross that unknown goat with the homozygous recessive on purpose. Like if I were to have like, it could be this, or it could be this, I don't know which one it is. All I see is it shows the dominant trait. I cross this question mark with a homozygous recessive on purpose because I can also see the recessive trait and that one doesn't have any weird questions. Like it's not questionable of like what the genotype is. Then you look at the offspring, which tells you which one it is. That's really what a test cross is. The 1-to-1 ratio is a heterozygous and a homozygous recessive cross of the test cross. So maybe, yeah, if it's confusing, just don't think about it as test cross. I just got used to saying that. Okay.
I mean, well, I mean, the test cross helped. I feel like it helps me think about it. But I was just saying, where'd you get the? I guess where'd you figure out that it is heterozygous at that point?
Oh, how did I figure that out? It's because of the 1-to-1 ratio of the long versus short. So we have 4 long goats, like 3 and then 1. That's 4 long goats. We have 3 and then 1 again for the short hair goats. So I'm just looking at whether it's long or short for like this B gene. So 4 to 4 is a 1-to-1 ratio, and a 1-to-1 ratio is always this cross. Like that, there's no question in that. But how you get that is like you could draw like a few examples of Punnett square that could be helpful. Like that is visually pleasing and easy to see, right? Like you can draw this much faster than I can like do the mouse thing. But if it helps you to draw it real quick, draw yourself some scenarios and see like what the outcome would be. Because if you draw any other Punnett square, you will not see the 1-to-1 ratio of dominant to recessive phenotype. This is the only way that you can have 1-to-1 ratio of the dominant to recessive phenotype. And it's because like this one parent, they can only pass on the little b gene versus this parent can pass on big B or little b, there's half and half chance, which then allows, wait, I didn't do this right, sorry. I didn't fill this in right. This one should be, yeah, which then allows you to have half the offspring with the dominant trait and half the offspring of the recessive trait. But it could be helpful. Like you can draw yourself a heterozygous cross monohybrid Punnett square and just have that available for the entire test. Like the first time that you see a genetic question, just draw it real quick, and then you can keep referring back to it if it shows up again. But maybe don't waste the time to draw it if there is not even a calculation question like this, because there are only 60 multiple choice questions and there are 8 units of a lot of information.
Gotcha. Thanks.
Yeah. Okay, so now we're on to non-Mendelian genetics, which is actually pretty straightforward. The notation over here is like whatever, you don't have to use that. Like you could do big A, little a, big A, little a. But if the question says it's incomplete dominance, then all you're figuring out is that the big A, little a is not showing just the big A trait, not just the red. It's gonna be a blend of the 2 colors, which is pink. So then in that case, if you're doing the calculations, then your calculations will be, like instead of a 3-to-1 ratio of the phenotype, it'll be a 1-to-2-to-1 ratio of the phenotype because they don't all have the same trait anymore. Okay, so that's incomplete dominance. Codominance is kind of similar, but codominance really can they can really play with it. They can obviously do the blood type example was like AB blood type, the genotype is IAIB. In that case, the antigen on the blood cell would have like the A antigen that would like we drew in class, but it would also have the B antigen on the red blood cell, like on the surface of the red blood cell. So that's the AB blood type. So that's codominance. There's not like A overcoming B, B overpowering A. They both overpower the O blood type, where like with the expression of the O allele, then there is no antigen of the ABO blood type on the blood cell. Yeah. Okay. So in this case, we have actually there's like one dominant one, there's this recessive and this recessive and this also recessive. But the question might say chinchilla coloration is dominant, or sorry, maybe it could like this really could be like a whole question. It could say chinchilla is dominant over Himalayan, but then black is dominant over chinchilla. So you could have like this, and that will be black. And then maybe you have like this, and that will be chinchilla. And then it might even say Himalayan and albino are codominous, in which case, if it shows up like this, oh, that doesn't make sense, does it? That doesn't make sense. Then it would just be white, because this one is just white. But like you could say, chinchilla and Himalayan are codominant, then you would have like both patterns showing up at the same time. But the question will tell you what is going on. You just like interpret it as it goes. Codominance just means both traits, both genes are expressed to the same degree, and both traits will show up at the same time. Okay, ABO blood type, an overview. I think we're pretty confident with this. Suppose, perhaps. Here's an example with this. It already tells you the genotype. What is this? Oh, they have 2 children. Both children are AB blood type. What is the chance that the third child will be blood type AB? First off, when you're looking at this question, you can say, oh, that part of the question is irrelevant because every child is a fresh child. The meiosis process goes through meiosis again. Fertilization happens once again, completely separate from all the other scenarios. So you would just draw yourself a Punnett square, find yourself the AB blood type situation, which is it's just 1 out of 4. But the question could get spiced up and say, what is the possibility that you have, just draw this real quick, like you can have A and then O, I just like IA, right? Like IO, A, and then O. So then you get, wait, no, it's a B. AB, BO, IBIO, IAIO, IOIO. So in this case, you could say, what is the chance that the first child has AB blood type? And the second child has B blood type. And the third child has O blood type. Well, now I got spiced up. Now you can do a calculation with this using the multiplication rule. Or maybe you can say, what are the chances that the first child could be either AB blood type or B blood type? In that case, it will be 1/2. So they can really ask you whatever questions they want. Let's answer. Toby doesn't like the sounds in the hallway. But there are a lot of sounds in the hallway. Living in the dorm. Okay. Toby just wants to tell them who's boss. And which is obviously not him, because he is such a. You don't look like a boss at all. You look so lame right now. Silly. Okay. What else over here? This is just saying like in your blood you have the antibodies. So if they clump, like if you have blood type A, you have anti-B. If you have blood type B, you have anti-A. If you have AB, you can't have any antibodies. If you have O, you have both. If you were to like, it's really important to donate the right type of blood, which is a red blood cell usually, to the right person. So like A can donate to itself, obviously, but A can also donate to AB because they don't have the antibodies. I think of the antibodies as a cop. So like they don't have anything out to get the red blood cells. So AB is the universal receiver. O is a universal donor because the blood cells are clean. RH is interesting. Toby, come here. Toby, why don't you sleep like Marshy? Marshy's just laying down chilling. RH blood type is like regular Mendelian genetics actually. So like this is positive, this is positive, this is negative. Exactly how this works, you don't need to know it, I would say. Okay, moving from there. X-linked. X-linked, we know how to do it. let's just look at a question real quick. X-linked could be recessive, it could also be dominant. So it's just a matter of like, is normal, like let's say it's a disease, is normal the dominant version or is normal the recessive version? Then the pattern will be different. So in this case, for X-linked, we have a wild type fruit fly, wide-eye fruit fly, and it's X-linked. So they gave you the notation instead of like big A, little a, they just say like wild type is using a plus. During the lab, we have a white-eye male. So there are two folds to it. You think, oh, it's a male, so it's X and Y. It's a wide-eye male, so it's XWY. Crossing with a homozygous red-eye female. Also, for the males, there's no homozygous heterozygous because there's only one. What we said? There's only one copy of the gene of an X-linked where'd it go? Of an X-linked gene for the males. But if it were to be Y-linked, it would just be all the males. But males do only get one copy from mom. Okay, so then we have homozygous red eye. We have in this case what? Red eye. So X plus, and then we have X plus. And then it says, then we have our first generation. So you got to draw yourself a Punnett square because it's just one Punnett square that's easy to draw. Draw that real quick. Toby, go to bed. Toby really is like really active today. He must have been sleeping all day. X, W, Y, X plus, X plus. So X, W, X plus, X plus, Y, X, W, X plus again. You notice that we have two female offsprings, two male offsprings. Okay, so it's asking us for the next generation, we're looking at a red eye female. So red eye female, we're referring to these. They're all red eyes anyway. So we're crossing another one of these with a red eye male. And they're all red eye as well. Oh, I wrote this backward. It should be like, it should be X plus XW because that's the dominant one. So you're crossing this with this. And then it's asking you what percentage of the, how many of the offspring are predicted. So you just draw yourself a Punnett square. You find yourself the wide-eyed male. Once again, we have the plus, the W, and the plus, and then the Y. So we get X plus, X plus, X plus, X W, X plus Y, XW, and then Y. We're looking for wide-eye male, so we're looking for the W one as a male, so we're looking for that one. So you use 1/4 times 5. Chinaza really is here so late. 1/4 times 573, and then we get that number, we round it to the nearest whole number, which is what it tells you. That would be your answer. And if it were to be, I mean, multiple choice, you choose the right answer. If you don't see the right answer, that means you got it wrong. If it were to be a free response question, write out the whole process in case you make a really tiny mistake that they might still give you the points for. You never know. So it's good to show the process either way. Okay, so that's this question. Ike's inactivation, you don't really need to know the details, but I'm just gonna mention it just in case.
Miss Bai,
Yeah.
for the last question, we didn't need to know which one was dominant.
Oh, good question. Great question, actually. Wait, oh, it tells you this one's recessive in this question.
Oh, okay.
Yeah. That's actually a good point. Like you bringing this up, let's just say it doesn't tell you it's recessive. Then that is similar to the test questions that we did for our unit test. So how do we do this? So if we don't know that wide eye is dominant, wait a minute. Hold on, this might be there's something wrong with this question? No, wait. Wait, I think this question is wrong, isn't it? I think red eye should be dominant based on this question. What did I do? Give me 1 second. So we did Wide-eye male, that's right. Crossing with a homozygous red-eye female, so that's right. But then the first generation females are red-eye. Ooh. Wait, if the first generation females are red eyes, that means red is dominant. This question is wrong. So red should really be the dominant one. Good catch. Okay, so this question like supposedly told you that wide eye is recessive. Oh wait, yeah, no. I read it, yeah, I read it backward. Wide eye is recessive, red eye is dominant. Yeah, that's fine. Yeah, red eye is dominant. That's fine. Yeah, we're good. We're good. But I was saying, even if it doesn't tell you wide eye is recessive, red eye is dominant, you still will be able to know that red eye is dominant because all the female offspring that are receiving an X chromosome from each of the parent, they're showing red. Like if all the offspring, female offspring, are showing the red coloration, then that means white is the dominant one and red is the recessive. Looking at the male is useless because the males only get whatever the mother has, and mother is homozygous wide eye, so all the male offspring will be wide eye anyway, so that doesn't tell you any information. So this is actually, yeah, this will make a really good question on the AP exam. Other questions about this? It is definitely important to think about how like the Y chromosome always comes from dad, or the male, the parent, and the X chromosome for a male always comes from mom. The two X chromosomes, one is from dad and one is from mom. And because male offspring only gets one X chromosome, they have a higher chance of having a like having a. Is my bunny on? It is. It's not moving. The oh, why are you so annoying today? What is going on? Oh, I know why he's so annoying. I didn't get a chance to take him out, so he's like needs to pee and constantly. Anyway, I was saying, what was I saying? X-linked recessive trait shows up more frequently in males, like male pattern baldness or color blindness. They're all X-linked recessive trait, like color blindness is linked to the recessive allele of the color blindness gene. And because male only receives one copy, they don't get a chance to be heterozygous and still not be colorblind. You either just be colorblind or not colorblind. Versus females, like it's a 3-to-1 ratio of not colorblind versus colorblind. Potentially, if it was a heterozygous cross. Or really, there are three different types of females. It's either homozygous dominant, it's either heterozygous, or it could be homozygous recessive. So out of the three scenarios of the three genotypes is a 2 out of 3 chance of not being colorblind and 1 out of 3 chance of being colorblind. Obviously, the numbers should be like based on actual scenarios, but like overall as a population, that's what the number is. Okay. X-linked activation is saying when there are 2 X chromosomes for a lot of organisms. For some cells, only one of the X chromosome actually gets expressed. I would say this is not important. So that also like gives you things like calico cats. Multiplication and addition rule, we already talked about it. But we can look at one more example. Do you want to look at this example, or you felt pretty confident with like doing questions like this? Actually, this is kind of a monster of a question. Should we do it? Because it doesn't like it's not asking for one particular phenotype. It's asking at least 2 recessive traits, which is so annoying.
Yeah, we can do it.
Yeah, let's do it. Okay. Okay, let's see. So having at least 2 recessive traits, there are several potential outcomes. You can have like there are 3 traits, right? So it could be recessive, dominant. It could be, yeah, recessive, dominant, and then recessive. Or maybe you're looking at dominant, recessive, recessive, or you can look at recessive, recessive, and recessive. And if this shows up, this is what I would say. If this question shows up on the AP exam, skip it, come back to it later. If it were to be a multiple choice, if you have the time for it, because it's going to take you like so many minutes and you might miss some easy questions because you ran out of time. Ideally, you don't run out of time, but it could very much happen, and it does happen for a lot of people. Anyway, for this question, we look at one trait at a time. For the first trait, we're looking for a recessive scenario. So heterozygous crossing with a homozygous recessive, we know it's a 1-to-1 ratio for the P gene, right? So to get the recessive, it's just 1-to-1, so it's half the chance to have little p. We're looking for little p, little p. Then we're looking for the recessive again, so we're looking for a little y, little y. Well, in this case, we're doing a heterozygous cross, which is a 3-to-1 ratio of dominant versus recessive. So we're looking for the recessive. That means we're looking for the 1 out of the 4 options, like 1 out of the total 4. The next one, we're looking for a dominant trait. A dominant trait, this is again a in my head, a test cross. So heterozygous with a homozygous recessive. This is 1-to-1 ratio again for the I gene. So for this I gene, we're looking for the dominant one, but it's 1-to-1 anyway, so it's going to be 1/2. So for this particular combination of traits, you would end up with 1/16. Now we do the same thing again for our second scenario. So for the recessive 1-to-1 ratio, it has to be 1/2. For the dominant one, however, now we're looking for the 3 out of the 3-to-1 ratio. So that's a 3 out of 4. And then for the recessive scenario, it's a 1 divided by 2. So in this case, the answer is 16, 3/16. Okay, our third scenario, we have dominant first. Again, we don't care. It's 1/2. But we're looking for the recessive again from the y. It's 1/4. Looking for the recessive again, but it's 1-to-1 ratio. It has to be 1/2. We have again 1/16. Now we do it a 4th time. Recessive, recessive, recessive. 1/2 times 1/4 times 1/2 again, giving you the answer 1/16. Now we gotta add them all together. So we do plus this, plus this. So what is the answer? So like all these different scenarios will give you at least 2 traits, even though we are only looking at one child, like one offspring. But we're looking at what are the chances of having at least 2 recessive traits, right? So it's really like it could be this possibility or that, or that, or that, which is why we're using the plus sign. And Chinasa, you missed this earlier, but the and associating with multiplication and the or associated with plus, that's given to you on the equation sheet, but you just have to be able to interpret the question and know, is it an and scenario or is it a plus scenario? Anyway, so adding all the numbers up, we have 16. And the top part will be 2 plus 3 is 5 plus 1 is 6. We simplify it, 3 divided by 8. And we go to the next page and we hope the numbers match. And it does! Hooray! We got it right. Questions about this, or feel good about it? Also, Chinasa, I would say the best ways to practice this for unit 5 especially, just practice the old question either from the packets that we did in class before when we learned the unit, or practice the problems from the exam or from the unit test, which I can make available over the weekend if you want to look at those. I'm on duty this weekend. This unit really is like the more that you do it, the more comfortable you are with it, and you are good at it from when you learned it, but it's just been a while. Okay, we did this. Write out all the gametes. We did this in class. That should be easy. That's like the brackets, AA, and then just the little b's, and then big C or little c, and then big D or little d. For all these, you end up with 8 different scenarios for the gametes. Linked genes. We talk about this pretty in detail during class. Let's take a look at this page just real quick and see if you have any questions about what you're looking at. Like, do you understand this is the same kind of question as this, But in this is related to the video that we watched about like this guy learning about the flies and was like, wait, these numbers don't match. And this is what he figured out. Not like this exact notation, but like this is just the gist of it. Leave this here for a minute for you. Let me know if you don't understand what you're looking at or how to reconstruct something like this. Okay. Assume you're probably almost done. Let me talk about just one thing real quick. Chinasa, you also missed this part. And also for the other two, Akeem and Harris, we were talking about this, but we weren't looking at it. So right now it'll make a little more sense. I was saying earlier that if you're looking at one parent and how it passed down its gamete, you really think about the law of segregation, and the law of independent assortment does not go in play anymore. The law of segregation is just that these two chromosomes separate from each other and go into like different gametes. It's just in this case, because it's homozygous, every single gamete will end up with the same exact genotype with one of these chromosomes. So yesterday somebody was asking like, oh, we have the recessives, but there's dominant over here. Like, why are there 4 gametes for the egg cells, but only 1 gamete for the sperm cell? So I was trying to say earlier, with our review session, is that like meiosis is within one parent, right? So like it's just this parent going through meiosis, ending up with this particular combination, and this parent is going through her own meiosis. So you end up with like this combination or this combination, or it crosses over and gives you this combination or this other combination. So these are the 4 potential egg cell from one parent, whereas this other parent, because it's homozygous, is just going to give you one type of egg cell. But then during fertilization, that's when the two actually combine, which then gives you the parental versus recombinant. And the parental is without crossing over can still happen over here. It just doesn't change anything. So we still consider these two as parental and these two as recombinant. Oh my gosh, wait a minute. I was supposed to go, oh no, hold on. Um, I was supposed to meet her. I told her I could probably walk Toby with Dot at 8:30, but now it's late. Anyway, yeah. So this is recombinant versus parental. Another way to tell recombinant versus parental is just from the numbers. Bigger number, smaller number. The bigger number is always parental. Smaller numbers are always recombinant. This is only when it's linked. Like if you see the 3 to 3 to 1 to 1 ratio, that's just like that is just normal. It could be from the earlier question. Also with the parental versus recombinant, you can look at the trace, like the actual physical characteristic. You can see how this one looks just like the parents. This one looks just like the parents. Parental, easy. But it could like bamboozle you if we also have a parental generation and the parent like the original, not the F1, the original parental generation, if it were to be like instead of having the plus plus for one parent, sorry, uh, like, let's just say, like, instead of having, let's say you have this for one parent, homozygous dominant, crossing with completely homozygous recessive, then you're gonna see the result. It's all consistent parental, traces back to the F1, traces back to parental generation, and that's fine. But if it's like the other example that we did in class where it mixes and matches, where like the parental generation is this, and then you have this, then in that case, we don't have big A and big B on the same chromosome to start with. Big A and little b start on the same chromosome. Little a and big B start on the chromosome, the same chromosome. See how like this is very different from what it is here, where like the both wild type are together and the mutants are together. This one is like the equivalence of like the wild type and the mutant are together, and then the mutant and the wild type are together. So now in this scenario for that other question, like the one that we did in class, if you're just looking at the trait, you have to trace back to the actual parental generation and not just the F1 generation from the F2 generation. That kind of makes sense. So just do some practice problems. If you're like, I will say redo this question on your own. If you know how to do it, you're good. Probably redo all these questions. If you know this is what you struggle with, in our big packet from the genetics unit, there are tons of these practice problems. Just do them. And I think the answer key for it is posted on Veracast. Or if you can't find it, you can find me and we can talk about it over the weekend. We did this in class, we did this in class, we did this one in class, we did this one in class. Ooh, this is showing you double crossing over. We did not do this one in class. This is showing you that like, let's say the recombinant frequency of like, let's say A and B recombinant frequency is like 30%. And B and C could be like 10%. And then A and C technically should be 40%. But in reality, it could be smaller. It could be like a 37%. And that is because A and C are so far apart that it can double cross over and appear as if they did not cross over. If that makes sense. Right? Like if you switch and switch again, did they switch? Yes. Are the numbers going to show that? No, because it switched back. So then the numbers are really like, you know, it's not going to be an exact number if it were to be real data. If this doesn't make sense, let me know. Okay. Pedigree. Ooh, yikes. I don't know if I can talk. Let me see. Pedigree and Pfizer are our last two things. We can talk about this real quick. Pedigree, you should be able to determine autosomal dominant, autosomal recessive. So is the disease like the dominant one, or is the disease the recessive one? X-linked dominant, X-linked recessive, is the disease the dominant one, or is the disease the recessive one? Normal is not always dominant. Normal could be the recessive. Y-linked will show up always in the males only, and none of the females will carry it. Chloroplast or mitochondria is from mom only to all the children. So like if mom is shaded in, then every single child will be shaded in. Something like that, okay? Some examples to be able to tell whether it's recessive or dominant, no matter whether it's autosomal, or like autosomal just means it's not X-linked. It's on like your first 22 pairs of chromosomes for a human and not the last pair of chromosome of the like the X chromosome. So you know this is recessive because it jumped out. But if it's the other way around, where like in this case, the not shaded in jumped out. So that means not shaded in is recessive. Yeah, recessive because it can like reappear. This is also X-linked because so not shaded in is recessive. So this will be like X little a y and X little a y. And you can just try to work it out. Also there was that like way to kind of help you remember if there was a recessive daughter with a dominant father. So let's say we have this one, recessive daughter with a dominant father, then that means it's not X-linked because if it were to be a recessive daughter, it has to be this. But then to be a recessive daughter, the dad has to be this in this case, right? But dad is actually not carrying, like doesn't have the disease or the trait we're looking for. So this doesn't work out. So it has to be like little a, and this will be like big A, little a. And she will also be big A, little a. So these are just practice, see the pattern. You get more used to seeing the pattern by doing the practice. This is an example that we did in class for a quiz. Can we do it super quick? Let's see. Oh God. Oh my goodness. Ugh. I don't have enough time to go through this practice with you right now. Well, we can see like this is not showing up as often, so it's probably recessive. Is there a recessive daughter with a dominant father? Recessive daughter. No. So it probably is autosomal. Try autosomal, see if it works. If it doesn't work, try X-linked, see if that works. Heterotrophic hypothesis, you can just look at it real quick. What even is this? Oh, we were thinking maybe autotrophs didn't show up at, like first. It was the heterotrophs that showed up first during evolution. That's just a thing. Chi-square, you plug it in. We practiced this in class already. Then we're done. We're done. Look at us go. Okay. That is all. I'm gonna go walk my dog. Thank you all so much for your attention, for your time. Tomorrow we do have the practice exam, but the main goal is to get yourself ready by Monday. So whatever the outcome is tomorrow, it's just practice. Okay. Don't worry about the grades. Just treat it as practice. Okay. Well, thank you all again for being here. Extra credit will be rewarded. Oh, extra credit will not be rewarded if I don't write your name. I have Akeem. I have Chinasa for half of it. I have Harris for the whole thing. I will see you all tomorrow. Bye bye.
Thank you, Miss
Bye.
Bell.
Welcome. See ya.
Thank you.
Bye.